vertebrate embryonic development, only a part of the egg or the cleavage mass of cells forms the actual embryo, while other parts lying outside the embryonic territory develop into extraembryonic regions called embryonic or fetal membranes. Embryonic membranes are auxiliary organs which have arisen partly for protection of the embryo and more specially to provide for its nutrition, respiration and excretion until the independent existence is attained. We would be discussing first the extraembryonic membranes in chick. In chick, the presence of an enormous amount of yolk and embryonic life to be spent within a shell is correlated with the development of extraembryonic membranes. Original blastoderm is a small disc which spreads by peripheral expansion and eventually covers the entire surface of the egg. But only the most central region is directly connected with the formation of the embryo proper. All the remainder of the blastoderm is extraembryonic. These are mainly 4 types. They are yolk sac, amnion, chorion and allantois. We would be discussing the development of each of these in detail, starting with development of yolk sac. In reptiles and birds, the somatoplure and splanchnoplure develop from the periphery of the blastodisc. These usually spread peripherally over the yolk mass. Soon afterwards, the embryo undergoes series of folds which appear all around the body of the embryo. These folds are termed as the body folds. The extraembryonic splanchnoplure, that is splanchnic mesoderm plus endoderm, constantly spreads over the yolk mass and eventually yolk sac encloses the mass of yolk in a large measure. The yolk sac, however, not surrounds the yolk fully. A small passage is left on the ventral side for the embryo to absorb the remains of albumin at a later stage. Immediately with the formation of the yolk sac, the intraembryonic splanchnoplure is subjected to fold resembling with the more superficial body folds, that is, the intraembryonic folds. The intraembryonic folds gives rise to walled digestive tract or gut in the body of the embryo. The middle of the embryonic gut remains open to the yolk beneath. At this level, yolk sac is connected to the digestive tract by a constricted yolk stalk. Development of amnion and chorion. The amnion and chorion are developed jointly as upward projecting folds, the amniotic folds of the extraembryonic somatoplure. The amniotic folds are named according to their location. They are amniotic head fold and amniotic tail fold. The amniotic fold first appears as a transverse fold in front of the head. It is called amniotic head fold. It grows upwards and then bends backwards over the anterior end of the head and covers it as with a hood. Another fold develops behind the embryo. It is termed as amniotic tail fold. All these folds finally cover an embryo in 2 sheets of somatoplure. The inner somatopluric sheet becomes the amnion and the outer the chorion. The amnion consists of a layer of extraembryonic ectoderm on the inside and a layer of extraembryonic somatic mesoderm on the outside, whereas the chorion is made up of a layer of extraembryonic ectoderm on the outside and a layer of extraembryonic somatic mesoderm on the inside. The cavity between the amnion and the embryo is called the amniotic cavity. In between the amnion and the chorion is the chorionic cavity or extraembryonic xylem. Development of allantois. The allantois arises as a ventral outgrowth of the splanchnopleur from the hindgut on the third day of incubation. It slowly enlarges as hollow sac and expands inside exocoel. Its walls are formed of an outer splanchnic mesoderm and inner endoderm. The proximal part of the allantois forms a slender neck or the allantoic stalk with which it remains connected with the hindgut of the embryo. The distal part of the allantois expands and penetrates between the amnion and the yolk sac on one side and the chorion on the other side. By the middle of the incubation period, the allantois spreads all around the egg underneath the chorion. The mesoderm on the external surface of the allantois fuses with that of the chorion, forming a conjoined chorioallantoic membrane. Moving ahead, we would be now studying extraembryonic membranes in mammals. The developing embryo of rabbit and other eutherian mammals are provided with 4 fetal or extraembryonic membranes, namely amnion, chorion, allantois and yolk sac as in chick. But in marsupial and placental mammals, embryos depends on the mother for food and oxygen and elimination of its own wastes. The fetal membranes begin to develop while the contact with the uterine wall is being established. Different extraembryonic membranes of mammals develop in the following manner. Development of amnion and chorion. Amnion and chorion develops simultaneously as upward projecting folds of somatopleur called amniotic folds. 2 types of amniotic folds develop. They are named according to their location. They are the amniotic head fold and the amniotic tail fold. First of all, the amniotic tail fold appears. It grows over the embryo dorsally and then grows forward. Afterwards, a transverse fold appears in front of the head. It is known as amniotic head fold. It grows upwards and then backwards over the embryo. Lastly, the head fold and the tail fold meet and fuse together. The place of their final fold develops. First fusion is marked by a provisional connection called seroamniotic connection or seroamniotic rough. After the fusion of the folds, the inner and outer layers separate and the inner layer becomes the chorion. The further development is same as in chick. Development of yolk sac. There is very less or no yolk in marsupial and eutherian eggs. Yet, a yolk sac develops in their embryos, which points to their reptilian ancestor. In the mammals, yolk sac begins to form during early gastrulation. In the blastocyst stage, the hypoblast endoderm cells found in the inner cell mass starts to extend along the inside surface of the trophoblast and encircles the inner cavity of the blastocyst. The cavity of the blastocyst within the enveloping endodermal layer is known as the yolk sac. Development of allantois. The development of allantois in mammals is same as in chick. Now we would glance the functions of these extraembryonic membranes. Starting with function of yolk sac in birds and mammals. The function of yolk sac in birds is to digest the yolk and transfer the product of digestion to the embryo. In mammals, the yolk sac is very little or with no yolk. Function of amnion in birds and mammals. The primary function of amnion is to protect the embryo from desiccation, and it provides private salty to an embryo to float. Amniotic fluid serves as an efficient shock absorber. Function of chorion in birds and mammals. A part of chorion forms finger-like outgrowth known as chorionic villi that penetrate into the wall of uterus for exchange of substances between embryo and uterus in the body of the mother. Function of allantois in birds and mammals. In birds, cavity of allantois serves as an urinary bladder. In mammals, the original function of allantois as urinary bladder becomes altogether lost. The carbon dioxide produced by the embryo diffuses into the maternal blood and is excreted by the kidney of the mother. Thus, at the end, to summarize the session that we have talked so far, the extraembryonic membranes are protective membranous structure that appear in parallel with the embryo and play an important role in embryonic development. It consists of chorion, amnion, yolk sac and allantois. The chorion participates in exchange of gases between the embryo and its surroundings, whereas the amnion protects the embryo. The yolk sac is sole source of food containing yolk, and allantois stores the metabolic waste of an embryo. Thus, the developing embryo is able to carry on essential metabolism while sealed within the egg and the womb